Welcome back to this next video in which we are discussing the thyroid gland. Okay, so we're currently in the process of discussing the control over the synthesis and secretion of thyroid hormones by the thyroid gland, which is of course done by the hypothalamic-Pituitary-Thyroid axis. So we've just been looking at the anatomy of the hypothalamus, and what I have found is the two portions of the hypothalamus that are going to be extremely involved in the hypothalamic-Pituitary-Thyroid axis, which are the arcuate nucleus and the median eminence. Now these portions consist of neuroendocrine cells, which are just basically neurons that are going to release hormones, and they're going to release them into a special blood system, a circulatory system known as the hypothalamic-hypophyseal portal system. Okay, so the first thing I want to do is make sure that everyone is happy with the concept of a portal vein. Right, so normally the way the arterial system works is you have an arterial system. This then breaks down into capillaries, like so. So I'll draw the capillaries here. And then these capillaries reconverge, and then you'll have a vein. Okay, so that's the way it normally works. And then this vein will go and drain into bigger and bigger veins, and that's the venous system. If, however, the capillaries reconverge into a larger blood vessel, which then splits into capillaries again, like so. So here are more capillaries, and then the capillaries are going to reconverge into a vein. So this is the vein now over here, and this is the arterial system over here. Then this structure in the middle, this is now called a portal vein. So a portal vein then is in between two capillary beds. So you have an artery here splitting down into smaller and smaller arterial blood vessels, such as arterioles, which will then split into a capillary bed. The capillary bed will reconverge up to form a much bigger blood vessel. And if that blood vessel then splits down into capillary beds again, which then reconverge up to form venules first, then they'll drain into a vein, and then that will drain into the entire venous system. Then this blood vessel in between the two capillary beds, this is called a portal vein. Okay, so that's the important concept of a portal vein. Now, a portal vein exists between the hypothalamus and the anterior pituitary. So the anterior pituitary, it's going to have a capillary bed in it, which has blood flowing through it that has already gone to the hypothalamus. So the hypothalamus is going to have neuroendocrine cells which put in hormones into the capillary bed here. They will then flow through the portal vein to the anterior pituitary, and then the anterior pituitary is going to be affected by these hormones, and this means that the hormones aren't really going to end up in the systemic circulation because they're just going to be removed at this point and go and affect the anterior pituitary whilst they're at this very high concentration. So they will end up in the systemic circulation, but they'll be in tiny concentrations in the systemic circulation, whereas the anterior pituitary will see them at much higher concentrations than they are in the systemic circulation, because they arrive at the anterior pituitary long before they ever go into the main body of the circulation, where they're going to be hugely diluted down. Okay, and this portal vein system that exists between the hypothalamus and the pituitary then, it's known as the hypothalamic-hypophyseal portal system. So another name for the pituitary gland is to call it the hypophysis. So hypophyseal means pertaining to the pituitary gland. So the other name for the pituitary gland is to call it the hypophysis, like so. So hypophyseal just means pertaining to the pituitary gland. So it's the portal system that exists between the hypothalamus and the pituitary gland, and the hypothalamic-hypophyseal portal system. Okay, and I'll just underline this then in red here. Okay, so what's going to happen is the median eminence and the arcuate nucleus then are going to be releasing this hormone, which is a tripeptide. It's made of 3 amino acids, which is thyrotropin releasing hormone. Okay, and they're going to be releasing it into their capillary bed here, and it's then going to go through the hypothalamic-hypophyseal portal system to the anterior pituitary, and it's going to work on cells of the anterior pituitary. Now, the specific cells in the anterior pituitary it's going to work on are known as thyrotropes. So you need to understand the anterior pituitary releases all sorts of different hormones, and there are different cells in the anterior pituitary which are responsible for releasing the different hormones. The cells in the anterior pituitary which are responsible for releasing the hormone that is going to affect thyroid follicular cells are known as thyrotrophs. Okay, so I'll draw one of these cells here then. So here it is, a nice round looking cell like so. So this is going to represent then our thyrotroph. So what's going to happen then is the median eminence and the arcuate nucleus, the cells here are going to be releasing thyrotropin-releasing hormone, TRH, into the hypothalamic-hypophyseal portal system. It will be coming down to the anterior pituitary, and it will affect the thyrotrophs in the anterior pituitary. So the thyrotrophs are now going to respond. And what are they going to respond with? Well, of course, they're going to now release another molecule which is going to go down to the thyroid gland and tell it to release thyroid hormones. So they're going to release now thyrotropin, or it's also called thyroid stimulating hormone, TSH. So TSH stands for thyroid stimulating hormone, but as I say, another name for thyroid stimulating hormone is to call it thyrotropin, hence why the hormone released by the median eminence and arcuate nucleus neurons or neuroendocrine cells is called thyrotropin releasing hormone rather than thyroid stimulating hormone releasing hormone. Okay, so to summarize then, to get the thyrotrophs to release thyroid-stimulating hormone, you have the neuroendocrine cells in the median eminence and the arcuate nucleus of the hypothalamus releasing thyrotropin-releasing hormone, TRH, into the hypothalamic-hypophyseal portal system. This TRH is going to go down to the thyrotrophs in the anterior pituitary, and it's going to drive them to release thyroid-stimulating hormone into the bloodstream. The thyroid-stimulating hormone will go into the systemic circulation, so it will be put in here, and it will go into the veins here, and it will end up in the systemic circulation, and it will make its way round to the follicular cells of the thyroid gland. And at the follicular cells of the thyroid gland, it is going to be stimulating the synthesis and secretion of the thyroid hormones. So let me just go over what it's actually going to stimulate in the follicular cells of the thyroid gland. So here, remember, is the process summarized for synthesis and secretion of thyroid hormones. Remember the follicular cells, I'll re-summarize it because we did it quite a long time ago. And the follicular cells of the thyroid glands, they produce this protein thyroglobulin, and they secrete it into the lumen of the thyroid follicle. Then what they're going to do is they're going to iodinate the tyrosine residues within the thyroglobulin protein, and to do this they need a high iodide concentration in the thyroid follicle. Therefore they have these means, these proteins, the sodium iodide symporter and the pendrin protein on the apical membrane, to transport iodide from the basolateral extracellular fluid into the luminal extracellular fluid, so that we can build up iodide concentration in the lumen of the thyroid follicle. Okay, right. Then what's going to happen is the enzyme thyroid peroxidase, which is put into the apical membrane of the thyroid follicular cells, it's put into these secretory granules which contain the thyroglobulin, and then when those are exocytosed, the thyroid peroxidase ends up in the apical membrane. That's going to catalyze the iodination of the tyrosine residues to monoiodotyrosine and diiodotyrosine. It's also then going to catalyze the coupling reaction, which will produce T3 and T4 amino acids, as well as dehydroalanine amino acids in the thyroglobulin protein. Then what we do is we endocytose the thyroglobulin, and we fuse the endocytic vesicle with lysosomes to form a lysoendosome here, and the enzymes within the lysoendosome are then going to break down the thyroglobulin to release the amino acids in there, and of course some of the amino acids that are going to get then released are these T3 and T4 molecules. Okay, so which part of this process is thyroid stimulating hormone going to activate? Well, all parts of it. So it is going to activate the synthesis of thyroglobulin, it's going to activate the synthesis of thyroid peroxidase, it's going to activate the synthesis of sodium iodide cotransporters here. Okay, so this process is going to go up. The transport of iodide into the lumen of the thyroid follicle, the production of thyroglobulin is going to go up. So production of thyroglobulin, the number of thyroid peroxidase enzymes in the apical membrane of the thyroid follicular cells is going to go up. In addition, it's also going to, all of those things are going to result in more actual synthesis of T3 and T4 in the thyroglobulin proteins. So we're going to end up with more thyroglobulin proteins with T3 and T4 amino acids in their structure in the thyroid follicle lumen. In addition, we're going to activate the endocytosis process here so that we are actually endocytosing more of the thyroglobulin with the T3 and T4 amino acids in them and breaking them down to release the T3 and T4. Another thing that I should mention is thyroid stimulating hormone also activates the thyroid follicular cells' production of hydrogen peroxide. And remember, hydrogen peroxide is extremely important for the function of the thyroid peroxidase enzymes. You need that for the enzymes to actually catalyze the iodination of the tyrosines and the coupling reactions. So we need to activate production of hydrogen peroxide if we want to increase our production of T3 and T4 residues within thyroglobulin proteins. Okay, so all of those, I think 5 things that I've mentioned there are going to be activated in these thyroid follicular cells by thyroid stimulating hormone. So overall, thyroid stimulating hormone acts on thyroid follicular cells to increase, or to drive rather, the synthesis and secretion of T3 and T4. Okay, right. So now let's summarize this all together and talk about the negative feedback control of the hypothalamic-Pituitary-Thyroid axis. So overall, the hypothalamus, the nuclei in the hypothalamus, the arcuate nucleus and the median eminence, they scream at the anterior pituitary via this molecule, thyrotropin-releasing hormone, to make the anterior pituitary in turn shout at the thyroid glands to release thyroid hormone. Okay, so the hypothalamus tells the pituitary to tell the thyroid gland to release thyroid hormone. Okay, now we need this system, in order for this system to actually control the secretion of thyroid hormone to make it match how much is actually needed, i.e. to maintain a constant blood concentration of the thyroid hormone, we need the hypothalamus and the pituitary glands to be negatively feedbacking. Well, we need the thyroid hormone concentration within the blood to negatively feed back on the release of thyrotropin releasing hormone and thyroid stimulating hormone by the hypothalamus and pituitary respectively. So what's going to happen is T3 and T4, they are actually going to go up into the thyrotrophs and also up into the neuroendocrine cells in the hypothalamus. So they go both to the hypothalamus and also to the pituitary, and they decrease the release of thyrotropin releasing hormone from the hypothalamic structures, so from the arcuate nucleus and the median eminence, and they decrease the release of thyroid stimulating hormone from the pituitary. Now you might think that T3 and T4 should just act on the hypothalamus, they should just reduce the release of thyrotropin releasing hormone, and indeed that would work, because of course reducing thyrotropin releasing hormone would then lead to a drop in thyroid stimulating hormone, but in actual reality they act on both of these. They act at both levels to reduce the release of TRH and TSH, and of course that's overall going to reduce the amount of TSH which is stimulating the thyroid gland, and therefore when you've got high enough T3 and T4 levels within the blood, you're going to get less synthesis and secretion of thyroid hormones by the thyroid gland. Now, importantly, remember that the brain, and indeed the hypothalamus is part of the brain, and the pituitary gland, they both have in their cells the type 2 5' 3' deiodinase. Okay? Different from the type 1 5' 3' deiodinase that the hepatocytes and the cells of the kidneys had, deiodinase. Okay? Now, what I want to finish with is talking about this thing that I promised earlier on, about how if you suffer extreme caloric restriction, so if you go on a diet, if you go on a very extreme diet, and the archetypal example, as I mentioned earlier, of an extreme diet is the Atkins diet, where you cut out carbohydrate and you just eat fats and proteins, but you hugely reduce your carbohydrate consumption. So you wouldn't, for instance, eat any refined sugar products. Okay, if you do that, everyone knows, it's a common popular fact that the result of this is that your basal metabolic rate goes down. So the rate at which your body uses energy is going to go down when you restrict your caloric intake. So caloric restriction, so you're eating less, your basal metabolic rate is going to go down. Now, how is this actually achieved? Well, the thing that does not happen is that T3 and T4 levels go down. T3 and T4 levels within the blood stay exactly the same even when you have restricted your caloric intake hugely. And what does happen is the expression levels of type 1 5' 3' deiodinases in the hepatocytes and in the renal cells goes down. So the number of deiodinases, the number of 5' 3' deiodinases of the type 1 in the hepatocytes and also in the renal cells is going to go down. Now, that means that the conversion of T4 to the more active T3 in the hepatocytes and the kidneys is going to go down, which means that the actual effect of the thyroid hormones in the hepatocytes and kidneys is going to go down. Now that means that both of those tissues are going to stop making as much heat. Remember, the T3 and T4 in the hepatocytes and the kidneys, that drives the production of that futile cycle, that drives the expression of the sodium potassium ATPases and most likely also the sodium leaky channels and the potassium leaky channels to create that futile cycle which generates heat. In addition, in the hepatocytes, T3 and T4 stimulate gluconeogenesis and glycogenolysis to liberate or mobilize glucose into the bloodstream. Now, if we have reduced type 1 5' 3' deiodinase expression in those cells, the amount of thyroid hormone stimulation that they're going to be receiving is going to be reduced because now the T4 won't be converted into T3, which is the more powerful form. So they will be receiving less stimulation even though the T3 and T4 levels within the blood has not changed. Therefore, you will be producing less body heat because that futile cycle in the hepatocytes and the kidney cells won't be activated as much, and therefore you will be using less energy. So indeed your basal metabolic rate will go down. So this is going to lead to your basal metabolic rate going down. But here's the really important thing. You might think, well, maybe this will lead to some disturbance of the hypothalamic-Pituitary-Thyroid axis, and maybe the level of thyroid hormones in your blood would therefore end up going up. But that's not what happens. Your thyroid hormone level remains the same, and the reason, of course, is that the hypothalamus and the pituitary use the type 2 5' 3' deiodinase. So even though this one's gone down, they still receive the normal amount of thyroid stimulation, because remember, the level of T3 and T4 within the blood hasn't changed. They still have their enzyme at the normal level. The type 2 5' 3' deiodinase is still expressed normally, so they will still be receiving the same level of stimulation by these thyroid hormones, and therefore you won't get them being activated to release more thyrotropin releasing hormone in the case of the hypothalamus, and release more thyroid stimulating hormone in the case of the pituitary. Okay, so to overall summarize then with regards to the hypothalamic-Pituitary-Thyroid axis, the T3 and T4 is going to act on the hypothalamus, and when it's high in the blood, it's going to be inhibiting the release of thyrotropin releasing hormone. It's also going to act on the pituitary, and when it's high, it's going to be inhibiting the release of thyroid stimulating hormone. When, of course, it's low, then that inhibition will be lost, and then they will be becoming active. The hypothalamus will be releasing thyrotropin releasing hormone, which will drive the pituitary to release thyroid stimulating hormone, which will then go down and drive the thyroid to release more thyroid hormones. in times of extreme caloric restriction, such as having a diet, type 1 5' 3' deiodinase goes down in, for instance, the liver and kidneys. This reduces BMR, but it doesn't mean that you're going to get more T3 and T4 being released into the bloodstream, because the type 2 5' 3' deiodinase, which is found in the hypothalamus and in the pituitary cells, that remains normal, expressed at a normal level, and therefore they will be acting completely normally, and they'll continue to maintain the T3 and T4 level with this negative feedback loop at the normal level. Okay, so with that then we will finish this video on the thyroid gland.